Good evening everyone. Please just confirm whether the audio and the video is clear or not. Please just confirm whether the audio and the presentation is clear or not from your end. Today is the sixth class in our course, right? This is the sixth class in our marathon series, and today we'll be completing the topic of spectroscopy. If you have seen the previous class yesterday, we have done the part 1 of spectroscopy, right, in which we discussed about the basics, and if you have not yet watched it, the link is in the description box, or you can check it out in the playlist of the marathons. Okay, so let's just wait for 2. Okay, so let's start off with our class. Students would join in the time being. So okay, so yesterday we have discussed about a lot of content. Before starting the class, if you haven't yet downloaded our application, make sure you download our application. It is available both on the Play Store and on the Android, and these are the various courses that are available with us, and we are about to start the June 2023 course, in which we'll be dealing with all the units, general aptitude and other sections. So if you wish to enroll, you can enroll in that. We have an extra discount running in the course, so you can enroll in there and get your preparation done in a smarter manner. Yesterday we have discussed all these things. Today we'll be starting off with the UV visible spectroscopy. If you haven't yet watched the basics video about the yesterday's class, make sure that you join in there and make sure that you watch that particular class for a better understanding. Let's begin. So let's understand about the UV visible spectroscopy. So UV visible spectroscopy, it is an analytical technique that measures the amount of discrete wavelengths of the UV or the visible light that are absorbed by or transmitted through a biomolecule sample in comparison to the reference or a blank sample. Now, good evening Mansi. Now remember one thing, if you all have seen the previous class, we know what are the two main things onto which our spectroscopy principle completely depend upon. It is the electromagnetic radiations plus the biomolecule, and the biomolecule could be the protein, carbohydrates, right? We have discussed all of these things. So the UV visible spectroscopy is dependent, or it is based upon what? It is based upon the absorbance and the transmittance that we have studied in the Lambert Law. We have discussed about this particular law in yesterday's class. Now talking about the applications of the UV visible spectroscopy. So it is used for the measurement of what? It is used for measurement of quantitative and the qualitative assay for the proteins and the DNA. Quantitative assay can be measured by measuring the absorbance at different wavelengths, and the wavelength, if it is 260, so the biomolecule that would be measured or that would be observed would be DNA or RNA, and at 280 we would be able to analyze the proteins. And also the region from 500 to 300 nanometers provide valuable information about what? About the prosthetic groups or the coenzymes. Now, what is a prosthetic group or what is a coenzyme? If you all remember that these are all the subparts of enzymes structure. So this is all from the biochemistry unit, and this is something that we discuss in our main structured courses. So if you wish, you can join in there for the complete study. Talking about the absorbance that is shown by the different amino acids at the near UV range. So if you look into a like normal graph, this is how the graph, or this is how we actually get the final results. Next is the fluorescence spectroscopy, right? So in this, what happens? In this, a beam of light is directed to a given biomolecule, and the electrons of that particular biomolecules excites further. They emit some sort of inflorescence that is detected by the detector. Now this is, we have discussed yesterday itself. In the ground state, the electrons are in the normal level, as in when they absorb a specific amount of wavelength, they would be reaching to the excited state. But this particular excited state is unstable. So after the electron starts to become very unstable, it starts to move towards its ground state, at which it would be emitting out some kind of some inflorescence, and this inflorescence or this fluorescence is actually, and we are able to analyze the final product that we have. Okay, next, talking about the applications of the fluorescence spectroscopy. So just a second, yeah, talking about the applications of the fluorescence microscopy. So they are mainly used for, now remember about this thing, very important, time and again it gets repeated in the CSIR. So it is mainly used for the amino acids tryptophan, phenylalanine and tyrosine, base pair derivatives, chlorophylls, fluorescent proteins, and for the organic dyes. Please remember all these things, very important. Okay, next we have IR spectroscopy, that is the infrared spectroscopy. See, we have already discussed about the basics of the complete spectroscopy. So you have to watch the previous class first in order to understand this class. The complete playlist is available to you. You can watch all the lectures. So in this class, we are just discussing about the types. Yesterday we have already covered the main basics, where we discussed everything in detail. Third one, we have the IR spectroscopy. So in this, what happened? In this, the incident light on the biomolecule is of the infrared region. So there, if you remember, there are various regions in the spectrum, and there is a infrared region. So if infrared region light is being used, so we will say it as the infrared spectroscopy. इसमें होता क्या है? It is useful mainly for determining the primary structure of the biomolecules. Very important to remember. In this, every functional group will have a characteristic wavelength at which they show the IR absorption. So we can say that we will be using the functional groups in order to get our final work done. So these specific vibrations or the rotation frequencies absorb the IR at specific wavelengths. Next we have the CD, that is the circular dichromism spectroscopy. It is used for the the main application is for finding out the secondary structure of the protein, and this is something that has been repeated time and again in the exams. Very important. Please remember about this, that circular dichromism is used for determining the secondary structure of the proteins and the peptides. For example, alpha helix, beta sheets, etc. Now in this, what happens? We'll be using a circular polarized light that is passed through the secondary structure of protein, and then finally the result is analyzed. So in this we usually measure the ellipticity shown by the secondary structure of the proteins. Now ellipticity of the different structural proteins of different biomolecules are fixed. So depending upon the information that we have and depending upon the information that we get from our samples, we compare them, and compare करने के बाद we get to understand कि okay, this is the main similarity or this is the main thing that we have to understand about our unknown sample, right? Now this is how the ellipticity of different secondary structure is all about. So this blue line would be showing the alpha helix, red line is showing the beta sheet, and green one is showing the random coil. You need not to go into the depth of this, do not worry, just it is just for the representation. Next we have the NMR spectroscopy, that is the nuclear magnetic resonance. So it is used for what? It is used for the tertiary structure of the protein. So please remember that for the proteins we have either the secondary structure or the tertiary structure. For secondary structure analysis we will be doing the circular dichromism, and for the tertiary structure the NMR, that is the nuclear magnetic resonance spectroscopy, is used. So in this you need not to go into these depth, you just need to remember about the applications. So talking about the applications of the NMR, first it is used for determination of 3D structure of biomolecules in their native states, then in the functional group analysis, and last is for determining the structure of the biomolecules, or we can say the structure determination of biomolecules. Very, very important. Please remember about all these things. These are very, very important. So we have discussed about what all things. First we discussed about, yesterday we discussed about that what is spectroscopy all about, right? Then we discussed about the how does or what are the factors onto which the spectroscopy is dependent upon. Third, we discussed about the Lambert law, that is how does we actually get to understand the absorbance, which is A is equal to ECL. Then we discussed about the various types of spectroscopy. Today we have discussed the various types of spectroscopy, right? What are those? First we have discussed about the ER, CD, NMR, UV, right? We have discussed about all of these, and in the exam we do get questions from this particular topic. So if you have any doubt, any problem, please ask, otherwise we'll switch to the questions for a practice session. Okay, any doubt, any problem, please ask from your end, otherwise we'll be continuing with the questions. You just have to remember about main things, and that is what are the basics, then what are the various factors. There are basically two main factors onto which the spectroscopy is dependent upon. First is the concentration of the sample, right, and second is the thickness via which we are actually working upon. So this thickness of the cuvette is taken into the consideration. Then we have discussed about the Lambert law yesterday in detail, and today we have discussed about the various types of spectroscopy. Just remember each of the application, very simple, like I have given you all the applications over here, that IR is used for what, CD, NMR, UV. Make a list of it and then you're good to go for this topic. Any doubt, any problem, please ask. Please explain Beer-Lambert law. Okay, let's just quickly revise Beer-Lambert law. Yeah, see this, yesterday we have discussed about this as well, but let's just quickly recall it. And Mansi, even after this, if you find any difficulty, you can watch yesterday's class. Okay, see, what is Beer-Lambert law all about? So see, in spectroscopy, what is there? In spectroscopy, the biomolecules, we know that there are two main things onto which the complete spectroscopy is dependent upon, and that is that we have just discussed as well, that is the concentration, and second one is the thickness, right? So depending upon the concentration of the substance and the thickness, what we do, like for finding out the relationship between the complete thing, we have a Beer-Lambert law in front of us. So the Beer-Lambert law formula is this, that is A is equal to ECL. Now if you remember yesterday, what did we discuss? That if you want to find out sorry, if you want to find out about the carbohydrate content or the starch content or any other test that we do in the wet lab experiments, we use spectrophotometer, right? So in this spectrophotometer, what do we have? We have a cuvette in front of us. So depending upon the variety of the content that we have various types of cuvettes used. For example, for carbohydrate and starch, we are using the simple glass for the simple plastic cuvettes. But for toluene, what do we use? We will be using a glass cuvette. Why? Because depending upon the nature, we use different thickness of cuvettes. This is what is actually getting done over here. So for finding out the final absorbance, what do? We will be using the Beer-Lambert law, where E is the constant, that is the molar absorption coefficient. Then we have L. L is what? It is the optical path length, or we can say L is the, what do we say? L is the thickness of the cuvette that we are taking into consideration depending upon the variety of content that we have, and M is the molar concentration. So when you switch on the spectrophotometer, we get a specific value over there, that is our concentration, and when we apply the complete formula of Beer-Lambert law, we get the absorbance, that is the final quantity or the final amount of content that we have in a specific plant sample. Depending upon the variety of plants, the absorbance, the concentration, or we can say every single thing differs. So that is the reason why this Beer-Lambert law is taking into the complete consideration. So this was about Beer-Lambert law. If you have any other doubt, you can ask, and if it is clear, we can move forward with the questions. Okay, Mansi, is it clear to you now? Doubt clear? Great. Okay, so let's move forward with the questions now. We'll be discussing 2-3 questions so that you can get a good idea about it. We have discussed all of these things. Yeah, this is the first question for you all. Question is, the helix content of a protein can be measured by. Now if you have like completely understood the topic that we discussed, you can easily get the answer for this question. Question is, the helix content of a protein can be determined by using which of the following? Take your time, understand the question, and write down the answers in the chat section. Hello. Okay, Harshit. Okay, very good everyone. If you have learned, like if you have read about the question, like the complete class, the question becomes very easy. So let's do one thing, let's quickly revise all the applications so that we can get through the complete application as well. Real life application of biological processes. Could you please just wait for some time, like let's just get the work done first. Okay. Okay, let's do one thing, let's just quickly revise the complete application of all the things. Okay, just a second. Yeah, so let's see. See, this was the very first type we discussed, that was UV visible spectroscopy. So UV visible spectroscopy is used for what? It is used for finding out the quantitative and the qualitative assay of the proteins and the DNAs. So this cannot be the answer because we had to find what? We have to find the helix. Next we have the fluorescence. Fluorescence is again not used for it. Fluorescence is used for what? It is used for the amino acids, for the base pair derivatives, chlorophylls, etc. So fluorescence microscopy also cannot be the answer. Then we have infrared. So in infrared we are using what? For the infrared is used for determining the primary structure. Then we have the circular dichromism. Circular dichromism is used for what? It is used for finding out the secondary structure of the protein. That means for finding out the helix and turns. So the answer for the question that we have discussed, it is circular dichromism spectroscopy. Then we have the NMR. NMR is used for the 3D structure. So what is the answer for this question? Answer for this question is option number C. Is it clear to everyone? Like most of you got the answer correct. Is it clear to you? Yes or no? Yes, all of you who gave the answer, is it clear? Yes. Very good, very good. Now just a quick revision, write down in the chat section, the IR spectrophotometer is used for what? Quickly write down the answer, we have just discussed this, so it would help you to like completely revise it. Write down the application for the IR spectrophotometer in the chat sections, everyone. Quickly write down the answer. What is the application for the IR spectrometer? Harshit Sharma and this other student, please do not disturb the class. Please, I request you to not disturb the class. Criminal analysis, okay, very good Nisha. Mansi, other Harshit. The genuine students, please write down the answers. What is the application that we have just discussed for the IR spectrophotometer or the IR spectroscopy? Yes. So we have just discussed about all of these things. Then for the fluorescence and the UV spectrometer as well, make sure that you revise properly, make sure that you get all of these things done properly, very important. If it is done, so this was about for today's class. If there's any other doubt, any other problem, you can ask in the chat sections afterwards, in the comment sections. So this was about it. Make sure that you enroll in our new course, which is starting very soon from 9th of January for the June 2023 examination. It would be a full-fledged course for you all, where we would be discussing the theory, the part A, that is the general aptitude, and all the other classes, all the other topics as well. And most importantly, you will be getting the hard copy of the notes at your doorstep. So this is a new thing that we have initiated on our application. So it is available in very affordable price. You can download our application and get the same. Application is available both on the App Store and the Play Store. Get it done from your end. Thank you for watching this video. Do not forget to like, share and subscribe if you are new to our channel. Thank you for watching. Bye everyone. Take care. निशा, December 2022 yesterday एक update आया था, but it is not from the official site, so let's not rely upon that. Let's wait for the CSIR to officially give something. That is the reason why I have not uploaded anything, because there was a mail like circulating among students. So I would like suggest you from my end, do not believe anything till the time it is not given to you via the CSIR itself. Okay, so keep on preparing, keep on like practicing properly, and if you find any difficulty, I'm here to help you out. Okay, you can text me personally, you have the WhatsApp number as well, or you can download our application and stay connected there with us. Okay, Mansi, Nisha, Harshit. Okay, good luck everyone. Let's meet now. Let's meet in the next marathon very soon. All the very best, and wishing you all a very happy new year in advance. Enjoy your day tomorrow. It's the last day, and all the very best. Bye everyone.